All right, students, so this is the beginning of the module on influenza A, which is, of course, a virus. So we're going to be looking at the biology of viruses and how they respond. This is just the beginning. There's a couple of pictures of viruses there. These are the unit objectives. Make sure you spend a little time and look at these. I always encourage you to download these PowerPoints and maybe look at them as we go through them together. So what is influenza? And this is kind of what we're going to cover. How does evolution relate to influenza? How does evolution work on viruses? And, how does our immune system then respond to these flus? So, understanding the causes and impact of the flu is how we'll kind of get started. We're going to look at what is influenza. So bird flu, swine flu, H1N1, all of these all these are different types of flu, but they're all caused by a type of virus known as the influenza, which is where we get the word flu from. So, flu comes from influenza. We're going to look at what viruses are made of, what they look like, what they do to our bodies, and how they reproduce. And at the end, we're going to look at, how our vaccines produced. So viruses are basically parasites of our cells. Viruses are pretty simple things. They generally have some coat around them and a piece of DNA. I'm going to look at that. But what a virus has to do is it can't reproduce on its own. All right? It has to find some host cell somewhere. And once it finds that host cell, it can then kind of hijack the machinery of that cell to produce more copies of itself. Our cells have the machinery capable of producing protein, which is one of the things that the virus needs, and it also needs to make some genetic material. So viruses are made of a capsid, which is the protein, and the genetic material that they have. The genetic material can be DNA or RNA, and they have relatively few genes. They don't really have a lot of DNA because they're not complicated. They're pretty simple organisms, so they don't really need that many genes to do what it is that they do. Some viruses possess this thing called an envelope that surrounds the capsid. So we're going to look at this thing here. This is kind of this is a bacteriophage. This is a virus that infects bacteria. So viruses can also infect bacteria, and we refer to those as bacteriophages. So here we see the capsid, the DNA, and that's really kind of the main portion of the virus. Bacteriophages typically have a neck made of these little proteins and tails in which they can attach to the bacteria in order to inject their material. This is an adenovirus. It has a capsid surrounding the genetic material, and then you have these little kind of spikes or these little protein fibers sticking off of them. But some of these viruses have this external thing around them called an envelope, and it's the remnants of the plasma membrane of the host cell. And one of the things this envelope does is it helps to assist in the attachment of the virus to new cells. Here is an RNA. This is the influenza virus. Influenza viruses have these copies of RNA. They have a capsid surrounding it. So this is the capsid in here. They have an envelope, remember, made of a host cell membrane. And then they have these little spikes, and these little spikes help it to attach to new host cells. Well, how are these viruses named? There are several different forms of influenza, A, B, C, D. A and B are the two most common that affect humans. They're named by the WHO according to their host. In case it is not human, type A or B, strain, and then year of isolation. So if flu A is followed by a nomenclature on the types of spikes found on the surface, and they can also have lineage designations too that help kind of identify the history of the virus. So, for example, here are some viruses that were protected by the flu vaccine. You have influenza A, Hawaii 70, 2019, H1N1, PDM09-like virus. So if we look at this, you can see that it's named that it's influenza type A. It was first isolated in Hawaii. It's strain number 70. It was found in 2119. And this is the designation we hear most, which is kind of short form for A Hawaii 70 2019. So on the surface of influenza A, we also see these spikes sticking off here. Those spikes are typically glycoproteins, which is a protein with sugar. And it, of course, I've said it helps to assist in infecting host cells. And we can see here that we have H spikes and we have N spikes. And that's where the H1N1 name comes from. Well, our virus is alive. We use 5 characteristics to determine if something is considered living or not. These include the ability to process energy materials, maintain some internal environment, respond to stimuli, reproduce, and adapt to changing environmental conditions. And we already know that viruses are not capable of processing energy and materials by themselves, so they have to have help here. The virus can maintain an internal environment. This is kind of questionable if they respond to stimuli from the environment, but they definitely need help to reproduce, and they do adapt. So they meet some of these, but not all of them. And since they don't meet all of them, then we do not consider viruses living things. Historically, we've never called viruses living things, but this idea is being challenged by the discovery of a new group of viruses called giant viruses, and you can see they're very, very large. So do these quick checks. These are a good way to kind of check your understanding of what we've been talking about. So how do viruses reproduce? Well, they don't reproduce on their own, so not on their own. They have to have help. All living organisms use DNA as their genetic material as far as we know. We haven't found any bacteria or higher organism that uses anything other than DNA. And the sum of all the genes in an organism, we've called a genome. So we have a genome of about 46, we have 46 chromosomes, and that would be our genome there. Most viruses are very specific in the type of cells they infect, and some viruses only infect certain species and only certain cells within that species. And one of the reasons is that the proteins that they have are complex, they have specific shapes, and those specific shapes help them to target very specific molecules on a target cell. So let's say that you have a cell inside your body. So this is you, and on the surface of your cell, you have these little receptors that are maybe triangle shaped. Well, along comes a virus that maybe has this little triangle shaped spike on it. This is not to scale, by the way. Well, this virus would fit really well into this receptor, and so therefore this virus can then infect your cell. Well, once the virus has entered the target cell, first of all, it has to attach. And attachment comes through the capsid bonding with the plasma membrane of the host cell, and then it will inject its DNA. Do you see the little DNA thread there coming down into the bacteria? And what happens is the DNA will enter the host. The DNA will then integrate with the host cell's DNA. So this is the DNA of the bacteria. The viral DNA attaches, and then it lives. The bacteria will divide, copy it, divide, copy it. And as this bacteria divides, we get new viral pieces of DNA. The bacteria will then begin making viral components, and the viral components are then assembled into a new virus. Those viruses will then release and go and affect other bacteria. So this is the lytic cycle: attachment, penetration, biosynthesis, maturation, release. And this image goes with these words there. In the lysogenic cycle, following penetration to the cell, so this is the lytic cycle here. We discussed the lysogenic cycle. It's a little different from the lytic cycle as it kind of skips right to the biosynthesis and maturation part. Well, RNA viruses are a specific type of virus. They present a problem for a virus because the host cell's protein synthesis requires DNA as the starting template. Well, if you're a virus here and your genetic material in here is RNA, then you can't, the host cell doesn't really read RNA right away. You have to start with DNA. And so RNA viruses contain enzymes that convert the viral RNA into DNA. And that's how viruses get their RNA. So you have viral RNA synthesis, and what they do is they convert this RNA into DNA, and then you can make a messenger RNA translation where you get protein synthesis and you build new viruses from it, and those viruses can then go and affect other cells. Well, what is the effect of the flu on our bodies? So the primary targets of the flu virus are epithelial cells lining your respiratory system, which is why your throat gets sore, runny, fever, and these epithelial cells, of course, are exposed to the environment all the time, which is one of the reasons that the flu is pretty contagious. So influenza viruses enter the respiratory system through the nose or the mouth. They can be picked up on the hands. And then if you touch your face or handle food, it gets into your system that way. It can have an effect on your digestive system, where you have loss of appetite, nausea, vomiting. You can have muscle aches, headache, fever, chills, and everyone that's listening to this has probably had the flu at some point in their life. Well, every year we hear about the flu vaccine, and what's in a flu vaccine? Well, there's salts, which enhances the ability of the immune system to detect the virus. Antibiotics, which prevents the bacterial growth during preparation of the vaccine. Egg protein. Newer vaccines that we have now don't use eggs during production, but will not have these proteins present. Formaldehyde, which inactivates any toxins from the viruses or bacteria. Gelatin, which stabilizes the vaccine. And then thimerosal, which is a preservative present in some multi-dose vials of the vaccine that kind of helps keep it from breaking down. Well, there are two different forms of vaccines used each year. The most common one contains 3 inactivated viruses. It's called a trivalent vaccine because it has 3 viruses in it. And then sometimes you have these quadrivalent vaccines because it contains 4 strains of the virus. Well, one question that we hear is like, how do we select these viruses to give in the vaccine? And what happens is scientists start about a year in advance. There's a lot of influenza monitoring stations, and they send samples to flu centers in 5 countries. And then they look at that data, they determine which of those flus has the potential to cause that year's flu outbreak, and they'll target strains that have been identified, and therefore maybe they can protect the most people that way. And the most common method of producing these vaccines is to inject the virus into fertilized chicken eggs, allow them to incubate for a few days. The virus will begin to replicate, and then the liquid portion of the egg is extracted. The viral particles are isolated, purified, and that's how we get those viral particles to make the vaccine. And what happens is you get injected with viral particles, not the actual virus itself, and so your immune system will recognize these particles, build up an immunity to it, and then if you ever get the actual flu, maybe you'll fight it off before you get sick. The new process that we used for like the COVID virus is called a recombinant process. It only uses a small portion of the flu virus. And you can read about that recombinant process there. And that is it for what is influenza virus.